neutralizing antibodies in case you're treating a viral disease. And then you inject that either for prophylaxis, which we're not doing, but there is a randomized control study looking at plasma, convalescent plasma for COVID-19 in healthcare workers that have been exposed to COVID-19. But what we are doing, and many people are right now in the US, is using it as therapy. So the difference between this and vaccines is this is passive antibody therapy, and vaccines are active antibody therapy. But for vaccines, as you will all know, you need to wait 3 to 4 weeks until you get an immune response and are protected from the agent you're vaccinating for. And sometimes it could be a little bit longer than 3 or 4 weeks. So plasma, the beauty of convalescent plasma is that you have it here and now when you need it in a sick patient, and you're not waiting for the vaccine to kick in. So the general principles is that looking at historical use of convalescent plasma, it seems to be more effective when you're using it as prophylaxis than as treatment. And the rationale or why is that? Possibly because when you get exposed, you have the inoculum of the virus or infectious agent is smaller than after you're sick, and that bug is multiplying in your body. So it's easier to take care of a smaller load of infectious agents than a big load. It's more effective when you use it early in the disease, and I'll show you some data on that. You need to give the right amount. The right amount not only depends on the volume of transfusion, but also on the antibody titers of that plasma. Not all convalescent plasma are created equal. Some patients develop a better antibody response than others. The historical sequence is that once you've identified that convalescent plasma works for the infectious agent you want to treat, then that evolves rapidly into companies producing concentrates of that plasma, which is immunoglobulin concentrate. And good examples of that we have had in the recent or still have are rabies immunoglobulin, hepatitis B, IVIG, immunoglobulin. We have it for RSV, and we have it for varicella zoster virus. And then it should evolve to monoclonal antibody therapy. Those are synthetic, those are probably more specific, and probably have less side effects. And interestingly enough, there is in the United States a research group that has genetically engineered cows in which they can make these cows produce human antibodies for a selected agent. And they're testing it for several diseases, and I think now they're working on COVID-19. So what is the timeline of passive antibody therapy? So it really had its success story in the late 1800s, early 1900s, when we did not have any antimicrobial agents, and it was used for several diseases back then with really good results. Actually, the first time it was used successfully was for a diphtheria with a plasma against the toxin of diphtheria. And that actually got the discoverer of that the Nobel Prize. And for 30 or 40 years, it was the mainstay of therapy for many infectious diseases, including pneumococcal pneumonia, pneumococcal meningitis, streptococcal diseases, etc., until the 1940s and late 30s when we got antimicrobials like the sulfonamides, then beta-lactams, penicillin. And when that happened, convalescent plasma or immunotherapy lost a little bit of headway, and it was not used so often until the 1960s when specific immunoglobulins for specific diseases was developed, and it had a little bit of a resurgence there. In the 1970s, monoclonal antibody techniques were developed. And despite the fact that they would be ideal for many infectious diseases, the reality is that most of them are used for autoimmune diseases. And there's only one that I'm aware of, or two, that are for infectious diseases. One is for RSV, which is palivizumab, and the other one that's in development is for Clostridium difficile. So that's kind of what's happened in the history of passive antibody therapy. So how does it work? So the antibodies have many different ways of working. First one is they can opsonize bacteria. The antibody attaches to the bacteria, and that lets your cellular immune system be able to engulf it, react better, and kill that bacteria. It could activate complement, and that by itself can lyse bacteria, or it could be directed against not bacteria but viruses directly, or the toxins produced by certain infections, like bacterial infections, like C. diff, for example, or diphtheria, and it just neutralizes the agent. Again, it could help the immune system produce, the T cells directly, T killer cells directly go and destroy the bacteria, and it can have direct antimicrobial properties, and so on and so forth. So these are all the mechanisms in which antibodies help you combat infectious diseases. So regarding COVID-19, what is the hypothesis of how may they work? And there's two theories. One, that remember that the virus has this surface protein called the S or spike protein. This is the protein that many of the vaccines today are being developed. Excuse me. And that the antibody can attach to this spike protein and block the interaction between the virus and the ACE2 receptor. That's how it starts the first step in getting into the cell. Or there are host proteases in the membrane of the cells that are important in changing the structure of the S protein when it binds to the ACE receptor, so it can internalize into the cell. So this could be an alternative mechanism or where the antibody could act. At the end of the day, what you're preventing is that virus coming into your cells and multiplying. So, this is the list of, and it's not a comprehensive list. It's not a full list of all the infections in which antibodies have been used, but it's a pretty complete one. And the red arrows mark in infections in which we still use antibodies, either to prevent as post-exposure prophylaxis or in the treatment of these diseases. And tetanus is one of them, hepatitis B, use immunoglobulins for moms who are infected with Hep B to the babies when they deliver to prevent infection. We also use it in rabies. We also use it in RSV treatment. There's a monoclonal antibody for that. And I'm not sure we still have the VZV immunoglobulin, but we did until a few years ago for post-exposure prophylaxis. Excuse me. So does this work? Do we have data that has worked in the past? And actually the best study was done on Argentine or hemorrhagic fever. Some of you may know this as Junin fever. Junin is the place in Argentina, the area, the city actually, that this virus affected. And in Argentina in the 1970s, they did, this was the first randomized controlled study using convalescent plasma against a viral infection, I guess any infection for that matter. And what they found is that individuals who received the convalescent plasma had a 1% mortality versus 16% mortality in the placebo arm. And then there's a meta-analysis of the use of convalescent plasma in Argentina with this virus for the last 15 or 20 years. And when that meta-analysis shows that the mortality rate now is less than 2%, and back then when you get all the studies together, it was almost 40%. So this is probably the best study in convalescent plasma in an infectious disease. Why did they go to all that trouble in the 1970s? Because that had a great impact in the wheat production in Argentina, which was the mainstay of the economy of the country. So everybody got worried, and they did this study to protect farm workers, to treat them. Going back a little bit further, we don't have randomized control studies, but we have some really good case control studies and cohort studies. This goes back to the 1913. This was the use of antibody against pneumococcal meningitis, and they injected the antibody in the spinal canal. Here there's a study of 1,200 patients with a 30% mortality. And when the patients with meningitis were injected the serum on the first 3 days of infection, the mortality was 18%. When it was later than 7 days, 36%. So not only did this study show that this apparently works, but that you need to give it early. That's the important message. This was a similar study done in Greece, same deal, pneumococcal meningitis, and amazingly, they had very similar results. When they injected the serum intrathecally, 13% mortality versus 47% when it was after day 7. Also, there's a lot of anecdotes and cohort studies on pneumococcal pneumonia, and Dr. Cecil, which was the editor of Cecil Internal Medicine book, the comment was, it is fundamental principle in all serum therapy that to obtain best results, you must give it early. And I am making an emphasis on this point because when I show you the COVID data, that becomes a relevant piece of information. So this was a meta-analysis on data from the Spanish influenza 1918. And here, basically, this is the summary of all the studies. And you can see that mortality was decreased in those individuals who received convalescent plasma. And actually, it was decreased in all the studies, but when you pull them together, you also have a favorable result in decreasing mortality. And in the same studies, when they looked at early versus late treatment, there was also a significant, statistically significant that when you gave it earlier, it was best for the patients to decrease mortality. So it did seem to work in Spanish flu. Then we have this in the 2009 H1N1 pandemic. And again, I'm not showing all the data, but the treatment of H1N1 2009 infection with convalescent plasma reduced respiratory tract viral load, the cytokine storm, and also decreased mortality. And just for the sake of time, there's data on SARS-CoV-1 or the SARS epidemic we had in 2003, and it also showed beneficial effects of convalescent plasma. So does convalescent plasma work for COVID-19? So there's only been 3 randomized studies. This is probably the largest one, and it was done in China. It included 103 patients. The problem with this study is that most of the patients got the convalescent plasma 30 days, medium of 30 days after they started with symptoms. But this is kind of too late. If you look at all the data I showed you, it really seems to work in some infections when you give it early. Here, that did not happen because of the design of the study. They did look at antibody titers within the plasma they were transfusing, and they had a protocol on the volume they were going to be transfusing. So that was, all those things were really good, really well done. And the study showed lower 28-day mortality, greater likelihood of hospital discharge, faster improvement, but none of it was statistically significant. There was a trend to it, but they could not prove a statistical significance difference between the placebo group and the plasma group. But when you look at the overall study, this is the improvement on people who got convalescent plasma or the control group. You can see at the end, 28 days, there's really not a difference. But when you look at people who had severe disease but not life-threatening disease, there seemed to be a difference that was statistically significant. For life-threatening disease, no difference. So basically, the difference between life-threatening and severe is the timing. These people have had the infection for less days than the people that started plasma when they were in an event later or at the end of the disease. There's been several observational studies or case control studies for COVID-19. The number of patients, the first number of patients is not very high. They all seem to decrease mortality, but again, these are not randomized controlled studies. But the data, when you look at all of them, it favors the use of convalescent plasma. But again, we have to be careful. We had the experience of hydroxychloroquine. When you do observational studies or case control, it seems to work in some, but when you go to the randomized studies, it doesn't work. So I got this slide from Dr. Joyner. He gave a webinar recently from Mayo Clinic, because Mayo Clinic has been actually the engine of producing and distributing convalescent plasma in the United States. So they looked at 7 studies. The first 3 were randomized control studies. The first one is the one I just mentioned, the one done in China. This one was done in the Netherlands, the third one in Iraq. The number of patients were not very high. None of them showed a statistically significant decrease in mortality. But when you pool them with the matched control studies and do like a simple meta-analysis, they found that the mortality was 40% less when you gave convalescent plasma compared to, I'm sorry, the mortality was 50% when you gave convalescent plasma. I'm sorry, let me start over. The mortality was 20% when you gave convalescent plasma and 50% when you did not. So, you know, the problems with these studies is some are not randomized, and the ones who are randomized, the volume of patients is not very large, and then the plasma was not given as early as you should give it if you really want to use this as therapy. So the Mayo Clinic started a program in April, which I really admire what they did. In 1 week, they put this program together, and they started distributing convalescent plasma through all the United States. Right now, there's 2,600 hospitals or sites that are using it. 11,000 physicians registered to use it, and they've transfused over 60,000 units. Individual patients got 60,000 units of plasma, and I'm sorry, 60,000 places were enrolled and 40,000 got the plasma. So they did a design, an analysis of this data. Now remember, this is not a case control study. This is just an open label expanded access program, and they included everyone that received plasma between April and July of 2020. And you were included in the study of the transfusion if you at least got 1 bag of plasma. Some patients got 2 or more units of plasma. So what did they find? So okay, let me go back one thing. The interesting thing is the objective was to explore signals of efficacy, and this is very important and interesting because this is not a randomized controlled study. So you are looking for what could tell me in all those patients who got plasma if plasma is really working. And they looked at 2 things. One was there any difference in mortality in the timing? And the second was there any difference in mortality in the amount of IgG the plasma had in the different patients? The hypothesis would be, if plasma works, probably people who got it earlier would die less. And if plasma works, probably people who had a better concentration of antibodies against COVID-19 would die less. That's what they analyzed. So they looked at 7-day mortality. Now remember, everybody here got plasma. There's no control group. 8.7% of those who were transfused within 3 days died, compared to almost 12% of those who were transfused after 4 days. So there was a difference there. In the 30 days, that difference was statistically significant. And when they looked at mortality with in relation to the IgG antibody levels and the plasma bags they were transfusing, the bags who had highest IgG levels, the patient has 8.9% mortality compared to low IgG, 13.7% mortality. And this was statistically significant. So these are 2 indicators that plasma may work, despite the fact that we don't have a randomized control study that meets all the criteria that we would like it to meet. Now, having said that, one more slide here. So safety, you know, we transfuse plasma for many things, and like any intervention we do, it has its risk, but the risk seems to be low. Of 5,000 patients that got COVID-19 antibody-rich plasma, there were 4 related deaths to the transfusion. So there could be a complication. It doesn't seem to be very high. And I think you have to balance that when you're offering to the patient, tell them, like, anything we give, there's a risk. You know, when you give a penicillin shot for syphilis, there's a 1 in 50,000 chance the patient will die of anaphylactic shock, but that does not prevent you from treating them if you think it works. And here the main problem is that we really don't know if this works. We think it works. But having said that, yesterday, or before yesterday, the FDA came out with this FDA EUA for convalescent plasma treatment for COVID-19 is now on hold because they analyzed the data that I just showed you, and they don't think it's strong enough to authorized plasma for convalescent plasma for COVID-19 treatment. But what this doesn't mean you can't do it. This means you cannot do it outside of a clinical trial or an investigational scenario. So the way that we're doing it with the Mayo Clinic, it is under an investigational situation. So we can continue to do it. But if you are in a clinic and you're not enrolled in this Mayo Clinic group, you would have to request permission from the FDA that you are doing a research study, and maybe you'll get approved, but you cannot do it like we give remdesivir, for example. So in conclusions, convalescent plasma may work for COVID-19. You know, the final word's not out there yet, but if it works, it's probably when you give it earlier and probably when you have high titers of antibodies in the plasma and you're transfusing. Seems to be safe. There are some randomized trials that are ongoing, and hopefully we'll get results from those. And if they're favorable results, the FDA will approve this therapy. And just remember that until we have an effective vaccine, this is the only immunotherapy we have available. And we are all putting our money that we're going to have a vaccine in a few months, but and hopefully we will. But there's always a possibility that the vaccine won't work, and we can't rely on the vaccine concept only. We have to look for other treatment pathways. And that is it.
Thank you, Dr. Mira. Sorry about earlier, my computer froze. Don't forget to sign in to claim credits. We'll go ahead and move on to Christy Duke. Christy. She must have stepped away. So actually, Emily, stop share screen and we'll move on to Brig Riley. Sorry about this, guys.
All right, I'm back. Can you hear me and see the slides?
Yes.
Okay, a little delay. So pre-COVID, I was full-time on HIV and Hep C, and now I am obviously been pulled like everyone else into at least some COVID time. So I'm going to go rather quickly to make sure I leave some time for Christy. Some of the things I'm going to fly through, this is a great thing I suggest. It comes out every Thursday. These guys are interviewed about 20 minutes long on what's latest in COVID. These are the two New England Journal editors. And some of the things they highlight that we know, but I think they always provide some insight into is, we're going to see some seasonal impact from COVID and flu as we enter the winter season. And they stress this isn't just a temperature-based thing. This is based on what it does to human behavior. Two main things they talk about, of course, is more people are going to be indoors with cold weather, and that many college students are moving across the country and starting their enrollments in other places. So that's going to have an impact. They also note that it's going to be hard to maintain physical distancing, perhaps, as more and more people just get seasonal flu and normal things. You've got more people with sniffles and a cold. Now physical distancing becomes even more difficult. Flu vaccination is going to be extremely important, and also respiratory diagnostics. So if someone has the sniffles, can we rule out COVID, and can we take them down a different path versus some of these COVID isolation protocols that are going to be required? So will we have access to timely and useful respiratory disease diagnostics? They made some guesses on where flu might go with COVID in the fall, and I won't get into those. They said it could be much better, much worse than expected, is all I'll say for now. They noted that it's quite frustrating that when you do a public health intervention in your community or at the state level, you don't see results for 1 to 2 months. Or if you fail to do something, you don't see the results for 1 to 2 months. So this is making sometimes sticking with the plan challenging and requiring more communications with stakeholders and the community. I just did a quick thing on where it's made the news. I'm going to let these titles serve as their own references. I'm guessing that you're seeing these probably in the Nashville area as well, as far as tribes have been harder hit by COVID. Every state seems to be coming out with that for their own tribes within their own state. Up on the top left, you'll see one on contact tracing, which is rather interesting and probably worth its own discussion. This one happens to take place in Arizona. And it wasn't true contact tracing, and I don't mean that in a bad way, but basically they would go to home visits of monitoring sick contacts who weren't ill enough to come in for the hospital yet. And that would often turn up a lot of household-based cases, and those people would be brought in for early oxygen therapy. So it was almost it was a household-based unit. And here's Cheyenne River making the news, flexing some of those sovereignty muscles to keep their reservation safe and keep their safety in place. Wanted to acknowledge, I think a lot of people are it's hard to keep this up for a long time, and it gets harder as we go on. As more data comes in, there's more pressures to, like, can we open up, or different pushes. So we can't really give people a timeline of when we're going to turn the corner, and I think that's increasingly frustrating for a lot of people. This is just from MMWR about a week ago that touches on this exact trend in kind of the national psyche. We're all suffering a bit from COVID, and a lot of mental health indicators are declining. And you guys talked earlier about sort of a lot of these things are just based on probabilities in individual situations, and that goes for community level too, not just individuals. So right now, casinos are trying to stay open, and schools are trying to go back. And so how do these fit into community level decisions? This is just going on longer than we had hoped, and the math keeps changing on us. I am gonna probably, well, I'll touch on this last statistic. I think it's worth noting. A new one has come out, but basically the one we care about here is that American Indian Alaska Natives are 5 times more likely to be hospitalized. Other data has come out with elevated incidence rates as well as death rates. This week's MMWR, if you're able to look at the one yesterday, American Indian Alaska Natives had a higher incidence rate. I think about 1.5% higher, but I'm not gonna game it here. It will be, I think, in a future echo. So this is really a reminder of the burden that the community gets, and the reference is there at the bottom. I think I'm going to go ahead and push past this. This is a large-scale diabetes study out of the UK. It basically reinforces the danger that diabetic patients have from COVID complications. What I will do is just kind of go to the slide. You can see the impact here. The red line is deaths among the National Diabetic Patient Registry in all of the UK for the previous 3 years. Averaged out, and then you see the green is how many patients died this year with COVID-related deaths, and the blue is non-COVID-related deaths. So basically, there's been a big increase in deaths among their diabetic patient pool, primarily, but not only, due to COVID. So it's been a huge impact in the UK. Again, bigger than perhaps what we've seen on a larger scale that's been documented prior. This was also seen in type 1 diabetes as well, not to this degree, but increased mortality. So I'm going to jump past this so I have time for Saliva Direct. This came out about a week ago, emergency authorization for a new type of test. And why do we care? We care because it uses saliva instead of respiratory swabs, so none of the brain ticklers anymore that have to go up the nose and that are very invasive. You don't want to do them too often, and you need a healthcare professional. So all of these limitations are gone with this new test. And there goes a train behind me if you can hear it. It's simpler. You don't need any preservatives at sample collection. You can have a sort of special collection cup. You can basically it is clear saliva. It does not. They do not recommend it for inpatients because often you'll have blood or mucus that will complicate things. So you want that kind of clear liquid spit, basically. They do not do nucleic acid extraction. This is part of the simplified procedure. They tested this over and over and they saw which steps they could remove to make this whole process simpler and without losing sensitivity. And they did this on a variety of vendors' platforms, as you'll see. So they checked with a lot of reagents, a lot of instruments. The whole point being here that it could be almost platform agnostic. Didn't matter what your lab was using. There were multiple ways up this mountain. And reducing the amount of procedures and reagents involved. So this would let us, in theory, do it at higher scale, less expensive. And so this would perhaps come in really handy if we want to test an entire set of school teachers or healthcare workers, right? You can do a large set of tests and get it done easily with a quick turnaround. This is from Yale, so it's open source. They're not commercializing it. It should be able to be done by existing labs with one big asterisk, which I'll get to in a second. And the estimate is going to be very cheap. The list prices have, if they did the math on these, and this should not cost more than $5 a test. So a big game changer in that respect. So they found 94% agreement with the most sensitive nasopharyngeal swabs, which are from Thermo Fisher. They found that it detected virus in a low limit of detection. And they found out how long you could store this without the sample degrading. So again, no preservatives, but it is stable for up to 7 days when the temperature stays below a certain mark. So again, collection and transport are very doable. Here's the schematic from the lab itself. This is very simple, but Dr. Mera looked at his lab. He's in a larger hospital, and it does take a certain level of certification, a high complexity certification. So it isn't just about having this dual-plex real-time quantitative PCR machine that goes ping. While the reagents involved in the process is pretty straightforward, only a certain lab is going to be able to do it. That said, I would think almost every one of our clinics, including those in Nashville area, are probably sending things out to Quest and LabCorp and this sort of thing. So I'd be very eager to hear which clinics, what you're hearing if you try to order this. Are the labs dragging their feet because somehow this is not a very commercial solution for them? Are they saying it's too hard? Are they saying it'll take 4 months? Are they telling you we don't know? So I think this will be a way where we can use our collective, our network to share information and figure out what commercial labs are getting it together or what problems they're throwing in our path. Because I think this could be a really important tool as we get into the fall season and perhaps want to test large populations in a short amount of time in a non-invasive way. So going forward, they still really much want to test this on a large population. As you know, with a lot of these COVID studies, it's often one descriptive study or a model or something like this. These they have tended to test on patients of about 50 to 60 on various platforms and reagents to check with its agreement with NP swabs. And so they have a pretty good alignment as far as sensitivity, but they still want to see what does this look like in an uninfected population? What does it mean when we find large amount of asymptomatic patients? So they still need a lot of numbers. They're optimistic that with automation, this can really move quickly. So you don't have to have somebody there with all the pipettes and doing this, or test pooling of the saliva samples, whereby you take a lot of samples and put them together. And if you get a negative, everyone's negative. If you get a positive, only then do you break out the samples and start to figure out who is positive out of there. So there's different ways that this could really increase our throughput. Again, I'm really curious to hear if our clinics are at the front of the line or at the end of the line, and how much the private sector avoids this or embraces this. This is their own words. By using many different vendors, we are not seeking commercialization. We're trying to make it as accessible as possible. As you know, if you're looking in a place with low prevalence and you're testing a lot of asymptomatic patients or just kind of doing a screening of the population, that's going to affect our false positive math. So these are the kind of things they're going to want. And in fact, they ask labs to join in and send them data, not data that should get anyone worried. It's about sharing the number of false positives and false negatives if they do validation studies. What's the next step? You don't buy this thing. You don't buy this thing in units. It is a protocol. So it's not a thing. It's a process that they are giving away that authorized labs should be able to do with existing equipment and existing reagents. Again, it requires a level of certification that we'll see how big of a barrier that is. So you need to be a CLIA certified diagnostic lab. You get designated as an authorized lab. They lay it out as quite simple, and I'll put the links there as far as next steps on what to do. So it's just a simple email. I think the main thing I'll do, this is the slide, this is the final slide and the one with kind of the important stuff. So the preprint on what they did and how they did it and the results is right there at the links. That's the manuscript. The protocol is the second link at COVID Tracker Connecticut, COVID Tracker Connecticut CT, and that will give you what data they want shared, where to email, and what to do next. I think most of our clinics, in fact, will not necessarily be doing that, but hassling their contract lab. And there is the copy of the emergency authorization at the bottom. And I think I shall leave it at that. That should be my last slide. If there's any questions.
So just to a quick comment, if I may comment, Brian?
Yeah.
So yeah, so just to reinforce what you said, I think this is a great test. It's cheap. It's you don't use swabs. It's just, you spit in a tube. And it's really not complex when you look how to set it up, but you do need to be a high complexity CLIA certified lab. And believe it or not, we have a big health system and we're not. So this is probably not going to fly for most of the labs in clinics, around the IHS clinics around the country. It might fly in a university lab or a high complexity lab, unless they modify this, unless they CLIA waive it. And I think that's probably the next step, is that they'll need to get a waiver for this. That's what I wanted to mention.
So is this available, sending it out through commercial labs?
So some labs are offering it. So yesterday, when I asked our lab director, can you do this? She said no. But then she started looking and there was this commercial lab that was offering. I don't know if it's this test in particular, but it was a saliva test that they were offering. And now I don't know what the price you're going to be asking, because this test costs about $4 a pop.
Okay.
We are paying at least 50 or 60 for a COVID test, and it's gone up to 250 in some labs. So it's very expensive. And like Brick said, we need more availability of tests.
So we had this question, Chief Francis from Penobscot Nation asked this question about the saliva test yesterday. So thank you. Thank you for that.
Yeah, and I think this is where when these clinics have asked whatever contract lab they've got going on, wherever they send out to, we're going to compare notes with multiple COVID echoes and try to see where there's blockages and where it's working, what we're hearing back. It's just so new, we don't know yet. So anything you hear from your sites would be helpful as well at this stage.
And one last thing, you did mention it, but this test is a little bit less sensitive than our regular tests. So it's not for hospital use. It's for outpatient use, massive scale outpatient use.
Great. Okay.
Would you recommend it at this point in time for asymptomatic screening?
Well, the short answer is yes, because we don't have enough tests out there. I'll just give you a quick example. I'm not a Native American, neither is my son. He needed a COVID test to go in. The university was requiring it. I'm an ID physician, and I spent like 45 minutes in the computer trying to find a place where he could get a test yesterday. And he was excluded for most labs because he was not symptomatic or blah, blah, blah. And I finally found one urgent care that did it, and the results were 5 days later. So that's the reality for your normal citizen in Oklahoma. And not everybody has 50 minutes to spend on just looking for a place to get tested.
So from a public health strategy standpoint, if I'm answering the question of a tribal leader, yes, but you know, a negative may not be a negative, and just be wary of that, and you know, don't clear that person just automatically.
Correct.
Correct.
Okay.
They did say there's some bridging protocols. So what that means is sort of if there's a different reagent or different quantity, Yale will give a protocol to basically to validate. So I could see a situation where they say, look, here's 100 tests, but could you do both a saliva and an NP? And we can see what the concordance is in your setting and with our lab. So that may be a phase that we go through as well.
Okay.
Okay, if there's no more questions, we'll move on to Christy Duke's presentation.
Okay, we can go ahead. I was going to say we could just skip it this week. I mean, that's fine. I give it every week. There are no dramatic changes in rates. I'll just touch on this very, very briefly. We're testing more. The number of positives are up, although Dr. Cotton reported yesterday that the number of new positives has decreased by 41% in the last week. I'm wondering if that's a testing issue, like Dr. Mira said, the shortage of testing, of tests out there, or if they're not testing asymptomatic patients. We don't know, but at least that's you know, a slightly encouraging sign. Over time, we'll know more, but there has been an overall decrease in the rate of new cases. You can see here, and again, we take this information from the state departments of health websites in the CDC. It is not AIA in specific, but we're tracking just as we have. The USET counties that make up the purchased and referred care delivery area versus the 13 states in which the tribes are located have been consistently higher than the US rates, and we're still tracking. It's still increasing. And we're tracking right along with it. It doesn't appear to be, we're not departing rapidly from the way that we're tracking upward. It seems like we're keeping pace for the most part, but we have been consistently higher. I expect that we're going to continue to see that as well. Again, we have the Redcap surveillance tool if you've not taken advantage of that. I know that we originally were going to be able to pay for that for a few months' time. We're going to be able to extend that. So if that's been your concern about utilizing Redcap for contact tracing and that sort of thing, just give us an email at usetepi@usetinc.org.
Thank you, Christy.
And that's all I have. Are there any questions for any of us before we hop off?
Just a reminder to fill out the survey in the chat to claim CEU credits.
Presenters can claim credits too if they'd like.
So no questions? Okay, everyone have a great weekend. Thank you so much.